In this video, we're going to talk about the second half of cell respiration, which is the electron transport chain, and that's all coupled with a process called oxidative phosphorylation. Okay, so just to review what we went over, the first two steps, glycolysis, we split glucose into two pyruvates. We get a total of 2 ATP there. That's called substrate level phosphorylation because we're making ATP by basically breaking the substrate glucose into smaller molecules. Then it goes into the Krebs cycle where we get 2 more ATP. That's also called substrate level phosphorylation. Okay, but then we have all these energy-rich electrons that are carried by these carrier molecules, NADH and FADH2. And in this last step, they're going to be passed from the NADH, FADH2 ultimately through a bunch of proteins in this electron transport chain to oxygen. So because oxygen is accepting them finally, it's called oxidative phosphorylation. It's a little bit different way to generate ATP, but it generates a lot of ATP because, again, oxygen is so electronegative, it'll accept these electrons even when we've extracted all of the energy out of them, and they're really kind of just low energy, tired, worn out electrons. Oxygen will still grab them and make water with them. So that's where we get that up to 34 ATP in that last step, this electron transport chain and then passing off to oxygen in this oxidative phosphorylation. All right, so let's get right to it. So hydrogen, again, is a reduced molecule. It's got these energy-rich electrons, and you could combine it with oxygen and have a massive explosion, right? That's what happened in the Hindenburg, right? We had basically its uncontrollable explosion. It released a lot of energy and it produced H2O, okay? Well, in your cells, we don't like explosions. We don't like uncontrollable reactions. We like controlled reactions. So we're going to take those hydrogens that have been extracted from food in the form of NADH, and we are going to pass them down electron transport chains, whereby at each little step we're going to extract a little bit of energy from them and use that little bit of energy to make the molecule ATP. When we finally have these low energy electrons, of which almost all the energy has been extracted, we're going to then combine them with oxygen. Again, oxygen is a very electronegative, so it'll take any old tired, worn out electron, even if there's no energy, and it'll accept it to make the molecule water, the final waste product of cell respiration. Okay, so let's look at that in a little bit more detail. So here's NADH and FADH2 being fed. These are a bunch of proteins, and they're passing the electrons down this transport chain. So this is basically an electron transport chain, and the electrons finally are accepted by oxygen to make water. Okay, well, let's look at this in a little bit more detail. What does this electron transport chain do, and how are these electrons passed? It's a little bit more complicated than this. So here's a slightly more elaborate version. Okay, so here's NADH passing the electrons from protein to protein to protein to protein to protein to protein. Each time, those proteins are using some of that electron's energy to pump the hydrogen ions that are also passed in up into this inner membrane space. This inner membrane space is out here. Okay, so as we are extracting energy from the electrons at each step, we're pumping these hydrogen ions from relatively low concentration out here to high concentration up here. Okay, and when we do that, we're actually creating a form of chemical energy or potential energy, because all these positively charged hydrogen ions don't like being packed together, right? So they want to get away from each other. So we're actually, it's like we're charging a battery. You can think of, again, putting all these charged particles together in one area in this inner membrane space. Okay, again, when we're done passing that electron, it's accepted by oxygen, and then the hydrogen ions that have shuttled back through here are going to be combined with it to make water. Okay, so where we get all the energy is all of these high energy, or all these hydrogen ions up here in high concentration, want to get away from each other. So we can actually then use that concentration gradient, the fact that they're all together and they don't like being around all these other positively charged hydrogen ions, to power them through. This is kind of like a turbine that's going to take that potential energy, and as they diffuse through here, that energy is going to be transferred to take an ADP and a phosphate and generate the molecule ATP. And this is where we're going to generate, again, most of the ATP within the mitochondria. This is the membrane, this inner membrane right here where this is occurring, by the way, right in there. Okay, so that's the last step, right? So we've got the electrons passed down this electron transport chain on this inner mitochondrial membrane. As they're extracting the energy from these electrons, they're using that energy to pump hydrogen ions into this inner membrane space. And then that potential energy is actually going to power the production of ATP. And this protein that they're funneling through is called ATP synthase. That just means it's synthesizing ATP. So that's the electron transport chain, and then this last step is oxidative phosphorylation, where we're using oxygen to accept the tired electrons, and we're using that to phosphorylate ADP into ATP. So that's the last step of cell respiration, where we get almost all the energy extracted from our food. Now, we have a problem though. I'm sorry. First, this is basically a close-up of that molecule ATP synthase, and you can see the high concentration of hydrogen ions here. Again, they want to diffuse from high to low concentration, so as they go through here, they can power the production of ATP from ADP and a phosphate. This is also called chemiosmosis, right? That just means chemical osmosis, not the movement of water, which is osmosis, but the movement of chemicals, in this case hydrogen ions, from high concentration to low concentration. So that chemiosmosis is powering the production of ATP. Okay, and this protein doing that is called ATP synthase. So again, a recap of this process. Glycolysis, we got 2 ATP, but we also extracted a bunch of these high energy electrons in the form of NADH. Okay, acetyl-CoA, we again extracted some more electrons in the form of NADH. Krebs cycle, we got 2 more ATP, but we got a lot more of the electrons, high energy electrons and their hydrogens, and then all these high energy electrons and hydrogens got passed on this electron transport chain, where the electrons powered the production of hydrogen ions into that inner membrane space, and then the hydrogen ions diffused back across through ATP synthase to generate all of that ATP in oxidative phosphorylation, where we generated about 34 ATP, okay? Total of about 38 for every glucose. That's a lot of energy. That's a pretty efficient burn, okay? But if we don't have oxygen, we can't do this last step, and we also can't oxidize all these electron carriers, these electron shuttle molecules here. So we have a problem, okay? And so this first step, this lack of oxygen, means we either can go in an aerobic direction if oxygen is present, or an anaerobic direction if oxygen is not present, okay? And the two types of anaerobic respiration we're going to talk about is ethyl alcohol fermentation or lactate fermentation, okay? And this is again because we don't have electron to accept those electrons in that final step, and because of that we can't do the Krebs cycle and we can't do the electron transport chain. We're stuck with just the energy we get from glycolysis, okay? So let's look at that in a little bit more detail. So here we go. This is alcoholic fermentation. So let's say you're a yeast cell and you're in a vat with a lot of sugar but no oxygen. Well, you can still take that glucose and you can, through glycolysis, get 2 ATP and produce pyruvate. Now you can't then send that into the mitochondria to oxidize it completely because there's no oxygen, okay? And in order for glycolysis to go, you have to have a continual supply of NAD so that you can continue to reduce that in the process of glycolysis. So what that means, unfortunately, is that we've got to take our pyruvate and convert it to another molecule, acetaldehyde, and then we have to actually take that molecule and reduce that into an energy-rich waste product called ethanol so that we can oxidize our NADH and produce NAD again so that we can continue the glycolysis process. So what we're doing here, unfortunately, is we're having to produce a high energy reduced waste product so that we can continue to recycle our NADH to NAD so the glycolysis can continue. And because there's still a lot of energy in these carbon-hydrogen bonds of alcohol, we haven't gotten a lot of energy out. We've only gotten 2 ATP molecules per glucose. So that's not a lot of energy for a glucose. But yeast can do this when there's no oxygen present. So that's what they give up. They give up efficiency for the ability to do it in an anaerobic environment, an oxygen-free environment. Okay, well, you don't produce alcohol when your body can't get oxygen, right? You get drunk, that wouldn't be so good, but you do produce another molecule called lactic acid. So your muscle cells can do this. Your muscle cells, when they can't get enough oxygen, will still go through glycolysis, turn glucose into pyruvate, right, and get the 2 ATP. Okay, that first step is the same, regardless of whether oxygen is present or not. Okay, but then muscle cells, if there's not enough oxygen, you're really running, you can't get enough oxygen to your muscles, they'll then take that pyruvate and they will reduce it into an energy-rich molecule called lactate. Why are they reducing it into an energy-rich molecule instead of oxidizing it? Well, because they need to oxidize the NADH back into NAD, so they're transferring these electrons back to this waste product, so that they have enough NAD to continue glycolysis. So because we're not using NADH in that last step, we've got to oxidize it in this second step of fermentation to produce a high energy waste product. And lactic acid, this is what makes your muscles feel sore when you're working out and your muscles start to feel that burning sensation. It's that lactic acid. Why is it being produced? Because you don't have enough oxygen in your muscle cells to completely oxidize pyruvate into carbon dioxide and water, which would not cause burning and problems. So these are two types of anaerobic fermentation that occur in different organisms when they cannot get enough oxygen to completely oxidize food in an aerobic way. And they get a lot less ATP per glucose molecule, but they can do this without oxygen being present, and they can continue to function in an environment that would usually cause them to die. Okay, so that's it. Final review of cell respiration.